Hello everyone, welcome to my channel Botany V4. In previous video, we have studied fungal septa. Today's video is about hyphal aggregations and modifications. As we all know that the thallus of fungus is unicellular or it is multicellular. So multicellular thallus is formed by aggregation of thread-like filaments called as hyphae, and these aggregation or this mass is basically known as what? It is known as mycelium. Now, in case of some fungi, you can see some specific macroscopic structures. You can see macroscopic structures, for example, fruiting body of mushroom, that is agaricus. So in some fungi, the macroscopic structures are formed by aggregation and modification of hyphae. And these structures are involved in different somatic and reproductive functioning. Somatic as well as reproductive functioning. So in this video, we are going to study total 7 hyphal aggregations and modifications starting from Rhizomorph, next Plectenchyma, Appressorium, Haustorium, Traps, Sclerotium to the last one, that is Stromata. So let's begin with first hyphal aggregation and modification, that is Rhizomorph. So in this picture, you can see that dark brown structures are there. These dark brown structures, they look like what? They look like roots. They look like plant roots, but they are not. These are what? These are all hyphal aggregations. So from this image and the word rhizomorph, you must have understood that this modification, it appears like that of root. So it takes the shape of the root. This rhizomorph word is derived from Greek words, that is rhiza meaning root and morph meaning shape. So these rhizomorphs are found in root invading or wood destroying fungi, and here several hyphae aggregate longitudinally lengthwise, and structure is so much so compact that individual hypha cannot be easily distinguished from this rhizomorph. So this picture is of rhizomorph of Armillaria melea, commonly known as honey fungus. These are the fruiting bodies of honey fungus, and this is the hyphal aggregation, that is rhizomorph. Now, if you take transverse section of this rhizomorph, the outline will be somewhat round, and if you observe this section under light microscope, what you will see is the peripheral region is dark in color and the central region is light in color. And if you take the longitudinal section of this rhizomorph, again you can see that there is one peripheral region and central region. You can distinguish two different layers, yes or no? The peripheral part and the central part. So here, a fully developed rhizomorph comprises of a central core and a rind or outer covering. The central core comprises of large elongated thin walled cells. Rind or outer covering is made up of what? It is made up of small thick walled cells. Now second hyphal aggregation and modification is Plectenchyma. So Alexopoulos and Memes mentioned that mycelium in most of the true fungi gets organized into either loosely or compactly woven fungal tissue, which is called as Plectenchyma. So Plectenchyma is of two types, Prosenchyma and Pseudoparenchyma. Now let's understand what is Prosenchyma. So Prosenchyma is a loosely woven tissue of hyphae. And as here the hyphae are loosely interwoven, you can clearly make out individual hyphae in this. And here these hyphae run more or less parallel to one another, and they are composed of what? They are composed of elongated cells. Next is second type of Plectenchyma, that is Pseudoparenchyma. Pseudoparenchyma is made up of compactly woven fungal tissue, and the fructifications of higher fungi, for example mushroom, agaricus, and you can also take an example of xyleria, dead man's finger. It is made up of what? It is made up of pseudoparenchyma. So here the first point is that in the fructifications of higher fungi, the hyphae become woven and intertwined into a compact mass. The walls of the hyphae in the mass gets fused, and hence they lose their individuality. You cannot find out separate or individual hypha here. The hyphal mass appears to be continuous structure consisting of isodiametric or oval cells, meaning what? As the hyphae are compactly woven, when you take cross section of this aggregation and observe it under compound microscope, you will find that these hyphae, they appear like what? They appear like parenchyma of plants. Yes, they appear isodiametric or oval. Now, as they resemble with parenchyma of higher plants, this kind of aggregation is called as what? It is called as pseudoparenchyma. Now, next, third hyphal aggregation and modification, that is Appressorium. This modification is found in case of plant pathogenic fungi. And Appressorium is the initial structure that is formed at the time of infection. Yes. So this is a special structure formed for attachment of the spore, or you can say fungus, to the host in early stages of infection. And here from this diagram, what you can see here is this is the uridospore, fungal spore, which is germinating. So at the time of germination, the spore develops what? A germ tube. Then the tip of the germ tube gets swollen. The tip gets swollen to form structure called as appressorium. So this part here is what? It is appressorium. And from this appressorium, one infection peg will grow, which will penetrate the host epidermal cell. Yes, so this modification is what? It is called as appressorium. And this appressorium can be found in parasitic members of order Erysipelas and Uridinales. Next hyphal aggregation and modification is Haustorium. What is the role of Haustorium here? The role is to absorb nutrients. And this Haustorium is found in most obligate and facultative parasites. Basically, Haustorium is what? It is a lateral outgrowth of intercellular hyphae. Yes, so if this is the mass here and this is the host tissue and this is the cell, host cell. So here one Haustoria develops that enters the cell. So this Haustoria, it is going to penetrate the cell wall, but it will not rupture the plasma membrane. Why? Because this Haustoria will continuously absorb nutrients from the host cell. And if the plasma membrane gets ruptured, meaning what? The cell is going to die and it is no longer beneficial, useful for this fungus. And hence, whenever the Haustoria are formed, they penetrate cell wall but do not penetrate plasma membrane. And this Haustoria can be found in various shapes or types, you can say, like simple Haustorium, branched Haustorium, and lobed Haustorium. So, if you compare a normal hypha and this modified hypha, that is Haustorium, what you can see here is, in case of Haustorium, the surface area is large. So if the surface area is large, meaning what? It can absorb more nutrients as compared to that of normal hypha. Next, hyphal aggregation and modification is trap. So traps are modifications of hyphae in predaceous fungi, which capture small animals, protozoa, rotifers, and nematodes. Now, some of these predaceous fungi belong to zygomycetes, but majority of them belong to deuteromycetes, fungi imperfecti. The fungi which capture their prey by adhesion possess no special modification of structure. Their hyphae secrete sticky substances, meaning what? These predaceous fungi, they can capture their prey by two ways. First is by secreting sticky substance. So their prey gets adhered, stuck to this sticky substance. And then the hyphae release what? Digestive enzymes like cellulase, protease, etc. And they take up the nutrients from the prey. So this is one method. Another method is what? Instead of secreting sticky substances, they create what? Traps for their prey. And once the prey gets trapped in these traps, then they are going to release those digestive enzymes. Yes. From this picture, what you can make out is a mechanical trap is made up of three cells. Yes, these three cells form a ring. And whenever the nematode passes through or crawls through this ring, the volume of cells in this ring, it increases by 3 times. And this is how the prey gets trapped. Now, these mechanical traps can be found in different predaceous fungi. The example here is what? It is Arthrobotrys. Next, hyphal aggregation and modification is Sclerotium. Plural is Sclerotia. It is derived from Greek word scleron, that means hard. So basically, Sclerotium is a hard resting body. Generally, it is round in shape, and this Sclerotium is made up of Pseudoparenchyma. Hyphal modification that we have studied earlier. The second hyphal modification that we have studied. Hyphal aggregation and modification. Yes. So Sclerotium is made up of pseudoparenchymatous aggregations of hyphae with or without addition of host tissue or soil. And this Sclerotium can resist unfavorable conditions. It can remain dormant for long time period. And whenever the conditions are favorable, this Sclerotium germinates to form hyphae or reproductive structures. Generally, this Sclerotium is produced in plant pathogens, for example, Rhizoctonia solani, Sclerotinia species, Claviceps purpurea. Now here you can see this sclerotium is of Claviceps purpurea, which has developed on the spike of wheat. And on the right hand side you can see here this is the sclerotium of fungus Sclerotinia sclerotiorum. Now the last hyphal modification, hyphal aggregation and modification is stromata. Please remember the term stromata. It is not stomata that is found in plants. Okay. So next hyphal aggregation and modification is stromata. Now, any fungal tissue that forms reproductive structures are called what? They are called stromata. And these are compact structures, somatic structures like matrices. So generally, when the sclerotium germinates, it forms stromata in which reproductive structures develop. So here you can see this is one example of stromata of Xyleria, commonly known as dead man's finger. And when you take the longitudinal section or even the transverse section of this Xyleria, you will find that the central mass is made up of what? It is made up of stromata, and in the periphery you can see fruiting bodies. This is an ascomycetes member, and the fruiting body here is what? It is perithecium. So in the periphery you can see many perithecia which are found as dark spots, and the central mass is what? The central mass is stromata. Yes. So here we have studied all these 7 hyphal aggregations and modifications, that is rhizomorph. Then next, second one, that is Plectenchyma, which is of two types, Prosenchyma, loosely woven mass, Pseudoparenchyma, compactly woven fungal mass. Next, third hyphal aggregation and modification that we have studied is Appressoria, which is developed at the early stage of infection in plant pathogenic fungi. Yes. Then fourth one, that is Haustorium. Fifth one, fungal traps, which are also called as mechanical traps. Sixth one, that is Sclerotium. And last, seventh one, that is Stromata. Now, if this video helped you understand different hyphal aggregations and modifications in fungi, don't forget to like, share and subscribe for more such videos. See you in the next video.